I'd like to invite Magnus to present next. His topic is also very interesting. It's about how microbiome affects cancer prognosis, which is really a surprising fact because people don't think of microbiomes often and how the presence of bacteria within our gut that is usually healthy can also diminish cancer and or also progress cancer. So, Magnus, you can start with it.
Well, is my screen visible?
Yes, Magnus, it's visible.
Welcome to second session of a webinar series. I am Magnus Andrew, scientific content writer at Scioverleaf. I'm here to speak on the topic microbiome and cancer, its implications and applications. Now there are two terms in this topic, microbiome and cancer. We all know what cancer is. Cancer is uncontrolled proliferation of cells and associated damage to neighboring tissues. According to National Cancer Institute, 2 million new cases have been reported in United States as of 2024, with 600,000 dying from the disease. The prostate, lung, and colorectal cancer accounts for 48% of the cases. In India, according to ICMR, Indian Council of Medical Research, it is expected to increase from 1.46 million in 2022 to 1.57 million in 2025. Now, what is microbiome? I think you all heard about gut microbiome. The microbiome is a collection of microbes such as bacteria, fungi, virus, and their associated genes that naturally live on our body and in the microbiome consists of 10 to 100 trillion of symbiotic microbial cells. The microbial community so high degree of variation depending upon the individual's diet, medication, genetics, and various environmental factors, and their imbalance, that is this biosis is strongly associated with diseases like inflammatory bowel system, Crohn's disease, obesity, and diabetes. Now what is the relationship between the microbiome and cancer? The Before that, we will see where all microbiome is present. It normally present in our skin, in oral cavity, in gut, especially in the colon, in respiratory system from the nose to the lung via the nasal canal. It also offers a unique microbiome, and in females, vagina said to possess a unique microbiome also. Now, what is the relation? The recent research have shown that the cancer harbors a unique microbiome like our gut, the oral cavity and all, and this microbiome is termed as intratumoral microbiome. This plays a crucial role in and modulating the tumor microenvironment and, and involves in tumor initiation, progression, and metastasis and immune response against the cancer. We will see how this microbiome influences tumor progression in both negative and positive ways. And at the end, we'll see how this microbiome can be used to treat cancers. Now, first, we'll see about the source of intratumoral microbiome. There are four pathways through which the tumor obtains its microbiome. The first is mucosal barrier invasion, where the microbiome present along the mucus layer can invade through the mucus and get transferred to the cancer present along the lining of the mucus layer. Next is hematogenic invasion, like microbiome present in oral cavity and intestine can associate with the cancer cells with freely circulating in the blood. That is during metastasis stage, the cancer the cancer cells can be freely circulating the blood, and the microbes can associate with freely circulating cancer cells and get transport to various sites along the cancer. Next is adjacent tissue invasion. This happens in case of oral cancer and colorectal cancer, where the oral microbiome and gut microbiome can easily associate with the cancer present in the oral cavity and in the gut. Next, tumor specific microenvironment. The tumor can also secrete substances which can attract these microbes from various microbiome present throughout the body. Next, how to study this microbiome? The identification of the microbiome involves traditional methods like culturomics, which involves the isolation of microorganisms from the cancer and subjugating them to 16S rDNA sequencing or multi-top mass spectroscopy to identify the species of the microorganism. It may also involve metabolomics, that is identification of specific metabolites produced by the microorganism, transcriptomics, the study of RNA expression by the microorganism, and genomics, which involves full genome sequencing. It may also it may also include immunological methods such as CLEM, correlative light electron microscopy, or FISH, fluorescent in situ hybridization for detection of microbiome or cancer. Now, how to study this microbiome? The study of microbiome involves in vitro experiments and in vivo experiments. In vitro experiments include cancer cell lines, study on cancer cell lines or spheroids, organoids, which are higher level of cancer cell lines, which mimics the natural cancer environment, or in vivo experiments, which involves cancer mouse models. Next, we'll see in detail about the effect of intratumoral microbiome. The cancer patient can be generally classified into two groups, the low risk group and high risk group. The studies in low risk group identify that they harbored a unique microbiome which are able to suppress the tumor growth. In case of high risk group, they harbor a different microbiome which able to able to induce tumor progression. The microbiome can prevent tumor progression by modulating tumor-related signaling pathways, activate T and NK cells, which are immune cells which play a major role in immunity against tumor, generate tumor-associated lymphoid system which mediate immune response against the tumor, the antigen presentation or secrete vesicles. It can also It can also induce tumor progression by upregulating immune checkpoints, inhibit T and NK cell activity, upregulating ROS, that is reactive oxygen species, and upregulating immunosuppressive cells which suppress the immune response against these cancer, or induce a pro-inflammatory response via cytokine. Cytokines are small proteins which play a major role in immune system. Like antibiotics, they are primary mediators of immune system. Next, we'll see about the major pathways which are involved in the effects of intratumoral microbiome. There are six major pathways: the toll-like receptor signaling pathways, nuclear factor kappa B signaling pathways, the string simulator of interferon new genes pathway, reactive oxygen species ROS pathway, and beta catenin signaling pathway, and tertiary lymphoid structure TLS production. Now we see in detail how intratumoral microbiome can prevent tumor progression. The intratumoral microbiome can activate the T cells, NK cells, which are immune cell mediating immune response against the cancer via stinging signaling pathway, for activate the production of tertiary lymphoid structures and and stimulate immune response by antigen presentation. It can also enhance the activity of CD8 plus T cells, which are cytotoxic cells, which have the ability to directly kill the cancer cells. And the tumor microbiome can also produce certain metabolites which have the ability to prevent tumor progression. This pathway shows some of the important metabolites produced by cancer which have anti-tumor properties, produced by microbiome with anti-tumor properties, such as short chain fatty acids (SCFAs). By activating various pathways, it can induce the production of anti-inflammatory cytokines such as interleukin 6, interferon gamma, and TNF-alpha, which mediate the immune response against cancer. There's also several other metabolites such as trimethylamine N-oxide, indole 3 acetaldehyde, indole 3 lactic acid, which also activate the production of anti-inflammatory cytokines which mediate the immune response against cancer. Especially the trimethylamine N-oxide has the ability to directly kill the tumor cells, releasing tumor-associated antigen and mediate the immune response against cancer via antibody production. Next, we'll see in detail about negative effects of intratumor microbiome. There are six major pathways to which the intratumor microbiome can mediate tumor progression. First is avoiding tumor immune destruction, where the intratumor microbiome deactivates immune cells such as T cell and NK cells, or induce the production of immunosuppressive cells such as TAM, tumor-associated macrophages, which prevents the immune response against the cancer. Next, it can also induce the production of pro-inflammatory cytokines via signaling pathways such as interleukin 1 beta, interleukin 6, thereby promoting tumor progression. It can also induce epigenetic changes in the DNA of the cells. Epigenetic modification is changes in DNA which which modifies the expression of protein, but it does not involve the change in basis of DNA. It can induce the microbiome can induce the DNA methylation, histone modification, and can induce chromatin abnormality, thereby upregulating oncogenes or tumor promoting genes, or tumors are downregulate tumor suppressing gene, thereby promoting tumor progression. Next is via genome instability and mutation. The microbiome can produce certain metabolites or toxin or induce the production of reactive oxygen species, leading to double strand breaks and mutations in the gene, thereby promoting tumor progression or tumor genesis. Next is tissue invasion and metastasis. The tumor microbiome have been proven to induce the metastasis of cancer, which is a transfer of cancer from one side to another, leading to spreading of cancer throughout the body. It can induce that metastasis by remodeling the chromatin structure in the cancer cells, thereby transferring them to metastasis cells. Next is metabolic regulation. It can the microbiome can upre- uh, lead to the utilization of glucose in large amounts, thereby producing lactic acid and other metabolites in faster manner, which can induce the which can induce the production of toxic metabolites or reactive oxygen, which can lead to damage in DNA and lead to cancer and mutations. To understand further how the tumor microbiome can influence the tumor progression, I have taken an example which is for Fusobacterium nucleatum. This is a gram-negative, non-spore forming, obligate anaerobic bacterium with slender shape and fusiform. It is a commensal organism largely present in the oral microbiome. Commensal means it does not have any negative effect on humans, but it has been shown that when it associates with cancer, especially colorectal cancer, oral cancer, pancreatic, breast, esophageal, and gastric cancer, it has shown to induce the tumor progression and metastasis via various pathways. It is the one such example where a single microorganism can associate with different types of cancer and induce tumor progression. Each microbiome, normally each microbiome have each cancer have a specific microbiome which influences progression. But when we analyze various microbiome present on various cancer, you will see a common microorganism which is Fusobacterium nucleatum. It is present in yeah, present in almost every cancer, every types of cancer, and where it influences the tumor progression and metastasis. Now we see in detail how the Fusobacterium nucleatum can mediate tumor progression. I will not go deep into this pathway. This there to show the complexity of the interaction between the Fusobacterium and the cancer. The first diagram show a generalized depiction of how Fusobacterium influences the immune response against the cancer. The Fusobacterium can bind to toll-like receptor 4 and activate nuclear factor kappa B pathway and lead to transition of macrophages from M1 type to M2 type. The M2 type is a immunosuppressive type, which means that it suppresses the immune response against the cancer. It can also lead to the apoptosis of TC cell, that is T cytotoxic cell, which are immune cell which have the ability to directly kill the cancer cells. It can also activate the product. It can also induce the production of various chemokines, which is also proteins which mediates the immune response, and cytokines, thereby which are involved in tumor genesis and metastasis. Or it can induce the production of inflammatory factors which activates immunosuppressive cells as like NK cells and T regulatory cells and lead to immunosuppression, or upregulate macrophages and T regulators and lead to chronic inflammation. Immunosuppressant chronic inflammation can induce the progression of tumor and metastasis. It can also induce the expression of various oncogene RNAs play a major role in tumor progression, such as via the same toll-like receptor 4 pathway. It can induce the expression of microRNA 21, which involve in production of certain enzymes which are involved in tumor genesis. It can also induce the progression of long chain RNA, which are involved in metabolic deregulation, and various other pathways which are involved in metastasis. It can also induce the progression of cancer by enhancing the production of transcription factors like the snail, slug, club, which are proven to be directly involved in metastasis. It can also induce cytokine reorganization, which are also involved in metastasis. Next, effect on colorectal cancer. In colorectal cancer, it can induce the tumor progression by epigenetic modification, the methylation, or to via its metabolites, which can lead to angiogenesis and tumor initiation, or it via beta catenin pathway leading to signaling which are involved in tumor progression. It can lead to apoptosis of lymphocytes, which are also immune cells involved in immune response against cancer, thereby promoting tumor progression in colorectal cancer. In oral, pancreatic, and breast cancer, first will in pancreatic cancer, it can lead to double strand breaks, uh, lead to production of inflammatory cytokine, producing inflammation, transition to M2 macrophages, thereby inducing the progression of pancreatic cancer. In oral cancer, it can induce the progression of tumor by immunosuppression or production of the certain factors which are involved in tumor progression. In breast cancer, it can lead to cytokine reorganization, thereby promoting metastasis. Now, what to do with this knowledge? The knowledge of the presence of intratumoral microbiome are being used to uh, used to treat cancer in variety of ways. Sorry for that. The knowledge that microorganisms have the ability to colonize on the tumor are being used to make the tumor targeting bacteria, tumor colonizing bacteria as uh, drug delivery drug delivery machines. Yeah, the tumor targeting bacteria can be loaded with the thermotherapeutic agents, immunostimulatory agents, nanoparticle, antigen, and chemotherapeutic drugs, which can prevent the tumor progression. There's also something called bacterial membrane vesicles. These are produced by a gram-negative bacteria during their growth, where the part of the plasma membrane cuts off from the membrane to form this vesicle. These bacterial membrane vesicles can be used at delivery machines to deliver the drug specifically to the cancer site without causing any systemic toxicity to the neighboring tissues. The tumor targeting bacteria can also be used for cancer diagnosis. If the tumor targeting bacteria can be coupled with any diagnostic agent and which can be used to diagnose cancer, it is a great help to treat cancer as the cancer does not get diagnosed at the initial stages. The major effects of cancer only shown when the cancer is fully developed, when it is hard to eat. So early diagnosis of cancer plays a major role in cancer therapy. In cancer diagnosis, I will provide an example. The scientists have used E. coli, which have the ability to colonize the colorectal cancer. They have they have genetically modified the E. coli to produce a compound called salicylate. Salicylate, which can be detected using normal methods. The proposed model is that when E. coli colonized the colorectal cancer, it induced the colorectal. The E. coli then produces the salicylate in large amounts when it associates with the colorectal cancer. This large amounts of salicylate can be detected in the blood and urine, so that the presence of salicylate indicates the presence of colorectal cancer. Now, how to improve the therapeutic effect and targeting? The scientists have used magnetic nanoparticles to guide the microorganism towards the cancer site. Uh, like I said, there are some microorganisms which prevent tumor progression. These can also be used as probiotic to prevent tumor progression. But to s- but to make sure that these microorganisms show their anti-tumor properties, the intratumoral microbiome has to be eradicated first, so that this probiotic can colonize the tumor more effectively. Scientists have also used to you increase the vascular permeability to allow that microorganism to easily colonize the tumor, and and seal these probiotics from the immune system so that they can effectively colonize the cancer. Next, we see in detail how the intratumoral microbiome can be used to prevent tumor progression. Uh, For this, the scientists have developed a probiotic which is lactobacillus rhamnosus and functionalized with gallium polyphenol network. The gallium have antimicrobial property and and able to kill the intratumoral microbiome. When this probiotic as administered orally, it can it found that it are able to kill the intratumoral microbiome, and the amount of lipopolysaccharide around the tumor decreased by fivefold. It also shown that the, the, the administration of this probiotic induced the production of anti-inflammatory cytokine, thereby preventing tumor progression. And the cancer is generally treated with combination with chemotherapy, radiotherapy, and immunotherapy. So, to test whether this probiotic treatment can be used alongside chemotherapy and immunotherapy, they have injected this probiotic orally and chemotherapy intravenously, and it was shown that combined probiotic chemotherapy approach have suppressed the tumor progression by 80%. In combination with the immunotherapy, the survival in the mouse models have been increased from 45 days when observed with immunotherapy alone to 60 days when this combination have been used. These results show that modulating the intratumoral microbiome can be a better alternative to chemotherapy and immunotherapy, and and this can also be used alongside chemotherapy and immunotherapy to prevent tumor progression. Thank you for listening to.